Welcome to Milestones, your daily snapshot of maternal nutrition breakthroughs. Today, Dr. Carla Cerami is joined by Professor Andrew Prentice to talk about nutrition and the early programming of the embryo.
Welcome to another of our podcasts on Milestones in Maternal Nutrition, part of the wider series of NNA Bites Big Learnings. I'm your usual host, Andrew Prentice, but today I'm going to be the guest to talk about nutrition and early programming of the embryo. So I'm handing my hosting duties to my colleague, Dr. Carla Cerami.
Thanks, Andrew. So I'm Carla Cerami, and I work alongside Andrew at the MRC unit in the Gambia, where I lead a research program of iron, infection, and anemia. I'm also Andrew's wife, but despite that fact, I still am going to have to read to you his biography, so I make sure I get it right. Andrew Prentice is a professor of international nutrition at the London School of Hygiene and Tropical Medicine. He lives and works here in the Gambia in the MRC unit, where he leads the nutrition and planetary health theme. And next year will mark the 50th anniversary of him starting his first postdoc position at our field station in the village of Kanaba in rural Gambia, which is actually where we're recording from right now. Andrew is a Google highly cited scholar with over 100,000 citations and an H-index of 136, which is very impressive, Andrew. He is a fellow of the UK Academy of Medical Sciences and was elected as an international member of the National Academy of Sciences, which is in my home country, the United States. He has been influential on many national and international expert committees and is very active in several academic societies. His work has been recognized by numerous international awards, which I'm not going to list all of them out right now, but I will tell you that the most recent one was the IUNS Lifetime Achievement Award, which he received in 2025. So, Andrew, before we get into the main topic, I want to start by asking you about your discovery science. And indeed, this is what brought us together over a decade ago when you hosted me and my team in rural Kanaba. And we did some pretty basic cellular science, elucidating mechanism by which iron supplementation increases the risk of malaria in young children and pregnant women. So it might seem strange to some of the listeners of this podcast that you actually focus a lot of your research on discovery science in such a remote setting. And there's many more immediate and practical issues here to be dealt with.
Thanks, Carla. Yes, we constantly have to sort of explain ourselves over this. So the first thing to say is we don't only do discovery science. You and I have done many randomized controlled trials, but we do believe discovery is important because for certain things in nutrition, we kind of know the answers and it's just about application and implementation. But there remain, to my view, a lot of the parts of the textbook that have not been properly written. And that's why the discovery is important. And I want to use an example of the work you and I have done together to really exemplify that, which is about the hormone hepcidin. So 20 or so years ago, we really were trying to work out why children responded so badly to iron supplementation. Then comes the discovery of hepcidin, which really opened new windows to us. And suddenly it became obvious that inflammation drives up hepcidin. Hepcidin positively blocks the absorption of iron in children or pregnant women who are inflamed. And that really turned around the way we understand the problem of iron deficiency. It turned around the way we might approach interventions. So that's one example, which I think is a really good one. Mm-hmm. And then as we go on through this podcast, you're going to hear really my favorite one, which I think really is going to open up new windows into future interventions.
Okay, so thanks for that. Let's get into our topic. Today, we're going to talk about nutrition and how it can affect some of the very first developmental processes in the early human embryo. But before we get into the details on that, on your work, can you just explain for the listeners why these first few hours and days of embryonic development are so important?
Yes. Well, first of all, to say that we've got this, what's called in the field DOHAD, Developmental Origins of Health and Disease. So this was work done, first of all, in the 1940s by Elsie Widdowson and R.A. McCants in Cambridge, and then brought to the fore by the work of David Barker. And that raised this acronym of DOHAD, Developmental Origins. We know that what happens to a baby in the first embryonic and fetal days is very important. It predicts lifelong health. What we don't know is what are the mechanisms by which these early effects are linked to later outcomes. Now, I'm constantly amazed by the thought of around the complexity of these things. Imagine sperm meets egg. You then divide into 2 cells, 4 cells, 8 cells. These are enormously complicated processes, and I'll say a little bit more about that in a second. And they all have to work perfectly. Otherwise, we don't get a lovely baby at the end of the process.
So would you like to tell us a little bit more about epigenetics, which I know is the focus of your work? But I think some people, including me, need a primer on this. So tell us first what epigenetics is all about, please.
Yeah, sure. But first of all, I want to call out my colleagues, Matt Silver, Prachand Isarapu, and various other colleagues who have really been doing this work. They're very smart bioinformaticians who can help us to understand what's going on in these immensely complicated processes. And I'm very privileged to be able to work with them. So epigenetics relates to control processes above and beyond genetics itself. So I think everybody will understand what genetics is. We know that every cell in our body has exactly the same constitution in terms of the genes, the nucleic acids in the DNA. And that raises questions. Well, if that's the case, if every cell is identical, how can you have different cells? How can we have a liver cell, an eye cell, a heart cell, or whatever? And the difference is down to epigenetics. So there are many different processes. We have methylation, where a methyl group is added to usually a CPG variant or a CPG site in the DNA. There are 30 million of these that can be adjusted. We also have microRNAs, histone modifications and things. And these all are really important in telling the genome how to express itself.
Okay, so yourself and Matt and the whole team have focused almost exclusively on methylation. Can you give us a little bit of information about why you've chosen to do that?
Yes, to some extent, it's almost an embarrassing admission, which is that people, including us, have focused on this a lot because it's been more tractable. It's been easier to do than looking at some of the other aspects, which of course some other people have been looking at, but that's the one that we've focused on. And that's what we're really going to be talking about in the rest of this podcast.
Can you give us a little bit more detail about what it is?
Yes, so I've already said that there are 30 million or so sites on the DNA which can be modified by the addition of a methyl group. Now, those sites are concentrated in areas of where genes are expressed, promoters, for instance, or repressors of genes. And I think a good way to help listeners understand this is to use a musical analogy, which is actually a very, very good one indeed. So think of our genes as the notes that are written on the staves of a piece of music. And think of the epigenetic marks as what we call the grace notes that are put on by the composer. So the composer may write pianissimo, fortissimo, quietly, loudly, or, faster, slower, etc. And those are the instructions that tell the player how to play that music. And the analogy is really good. They're the instructions that tell the nucleus how to express those genes.
Okay. I think one thing that's gotten lost along in this discussion has been, where does the nutrition angle come into this?
Yeah, thank you. So we need to produce methyl groups, what we call methyl donors, in order to have the methyl groups onto the DNA. So how do we produce that? Well, they're relatively complicated pathways, biochemical pathways that do this. And those pathways are highly dependent on a set of nutrients that we call the methyl donor nutrients. Those would include B12, B6, folic acid, riboflavin, the amino acid methionine, betaine, and a couple of others. So all of those come together, and if they're working properly, if there's sufficient of those substrates available, they'll allow the system to put methyl groups where it wants to on the embryo as it develops.
Now, what you just talked about was the methylome, and you described how it does occur mostly in the very early embryo. Can you just talk us through how that happens?
Yes, again, it's mind-blowing how evolution has devised such a clever process. So when sperm meets egg, the sperm carries its own methylation marks, as does the egg. And those are laid down, for the egg, they're laid down in about the 14 weeks prior to conception. For the sperm, they're laid down both in the short term and the long term. They're actually, some of those are laid down in the fetal life of the man who donates that sperm. And so they're dependent on his mother's nutrition when she was pregnant with him. So complicated processes. Now, what happens is when they meet is that most of the methylation marks have to be taken off in order to produce pluripotent cells that can then divide into liver, kidney, or whatever. And those are scrubbed off. They're scrubbed off the sperm very, very quickly and actively within the first few hours. They come off the egg more slowly and in a passive way as the egg divides, then not replicated. That's true for most of them, except for a subset of what we call imprinted genes, where the labels are left on in order that during placental and fetal development, either the mother's copy of the gene is used or the father's copy of the gene is used. That's a whole other story, which we could well do a podcast on, but I'll just leave it at that for the moment. So where we've got to is we inherit, if you like, from the sperm and the egg, some methylation marks. Most of those are washed off, but not all of them. And that's really important in terms of the possibility that you have intergenerational effects. And then they start to be laid back down again as the cells divide and specialize into the 3 germ lines, which then form all the tissues of the body.
Okay, so now let's get on to the incredible bit of serendipitous good fortune that helped you and Matt and the team make some important breakthroughs. And as far as I understand it, it's seasonality here in the Gambia. Can you tell us a little bit more about that?
Indeed. So we call this a seasonal experiment of nature. You and I are sitting here. It's very hot. It's the dry season. It hasn't rained for, 6 months now, and it won't rain again for another 3 months. So we get this huge seasonality, and that seasonality affects what plants are grown, what crops are grown, and what people eat. So what we did initially was to ask the question, can we see differences in methylation in babies who are conceived at the peak of the dry season or at the peak of the wet season? And lo and behold, we found that if you know where to look, and they're relatively rare regions called metastable epialleles, a concept too complicated for us to get into at the moment, indeed you find that some of these are highly sensitive to season of conception, and they have either a higher or lower rate of methylation. And I mean, to such an extent that we could almost, with some certainty, predict what month a baby was born in just by looking at its DNA, or even an adult was born in by looking at its DNA. So that really told us that the methylation rates are sensitive to some environmental exposures. Then what are those environmental exposures? Could they be malaria? Could they be different diseases? Could they be nutrition? We are not totally certain, but we have a high degree of confidence that these are related to nutrition and the different diets that mothers eat. And there's a story behind that, which again is a little bit complicated as to how we come to that conclusion. So I think we probably better leave it there, but more than a moderate level of security that this is about nutrition.
So if I understand you correctly, you're saying that the mother's diet and nutritional status before and at the time of conception could modify some of these relatively rare CPG sites, and that these sites are then replicated faithfully every time the cell divides.
That's an important point you've just made, that yes, once they've been changed, they stay like that, as far as we can tell, for most of a child's life. Now, that's different to some other sites which change when there are different exposures, such as smoking or high folic acid supplementation. So these are discrete areas that tend to remain the same so far as we can tell.
And they're also different from the epigenetic marks that mark the differentiation of cells into different organs.
Indeed.
I think that's an important point for other.
Well, here's another important point. Okay. Now you raise that, which I should have said really, that the beauty about these metastable epialleles is that they are laid down very early in conception, before what's called gastrulation, when the tissues start to specialize. And therefore, we can take a sample of any part of the body and we will get a representative measure of what's going on in the rest of the body, which is really useful in terms of the science we do.
Right. Okay, so now that we talked about that, Is there any evidence at all that these metastable epialleles affect health?
Yeah, well, of course, that's a crucial question, and one that is asked constantly of us by our lords and masters that give us our funding. Because, it'd be all very well to have fun looking at how a mother's diet changes these quite sort of discrete areas of the genome, but does it really matter? That's the question you're asking. And the answer is certainly it's going to. And we have quite a lot of evidence already. So some of the first work we did was related to obesity. And we can see that seasonally mediated differences in the methylation of alleles in the POMC promoter, which regulates appetite and hence obesity, are affected. The PAX8 gene, which affects thyroid function. We know that there are differences in growth and in pancreatic function, which I'm going to come back to in a second. Probably some cancers, notably through tumor suppressor genes. And what I would say is that these are just the first examples we've found so far, and there are almost certainly going to be many more, we believe.
Okay. And finally, you asked me to prompt you about something, which is the very latest work done by Matt Silver and his team, which I know you're very excited about. And it's something about what he's calling dark matter, which sounds strange, like it belongs in space research, not nutrition research. But can you explain this?
Yes. So, well, I won't explain it in too much detail because this is truly, truly complicated. But let's give a sort of high-level vision just to tempt people maybe to look into this a little bit more. So, intriguingly, if you look at the whole of the human genome, only 2 to 3% actually expresses genes. So what on earth is the rest of the 98 or 97% of the genome doing? Well, a large proportion of that is what's variously been called in the past dark matter or junk DNA. And it consists of some very ancient and some less ancient viral inserts. So when we've had a viral infection, as listeners probably know, viruses insert bits of DNA into our genome and then get us to do the work of expressing it. Now, some of those insertions have stayed there. They've stayed there for many, many thousands of years. And these are troublesome because they also move around. They're little bits that move around, often called jumping genes or transposable elements or transposons. And if they jump, if a little bit of section of DNA jumps from where it is to the middle of a genome or to a repressor or a promoter gene, then it can cause havoc. So the body has had to work out systems by which they can control this from happening. Now, intriguingly, although we've kind of known that stuff was going on here, it's been very difficult to research with great fidelity until recently. And the reason is that short-read DNA sequencing, it's very difficult to place because you're getting repeat sections that all look the same. So when you do short reads and you're trying to put them into the genome and reconstruct it, you really don't know where to put them. But now with the advent of long-read genetics,
you mean sequencing
genetics. So the long reads where you can take very long bits of DNA and sequence that. And we do that here in the Gambia using something called Oxford Nanopore technology, an amazing technology that allows you to read the whole of a human genome and including these long reads. So then you can really sort out what's going on with these jumping genes.
Interesting.
A question that comes to my mind is why are they left in the genome? Imagine the metabolic work that is being done every time a cell is replicating. All those 8,000 million or whatever they are DNA bases have to be faithfully replicated. Why are we doing this? Why don't we just kick these genes out and get them out of the system so that we could do much less work? Well, I think we still don't know the answer to that at all, but one suggestion is that some of them at least, may have been adopted to do some useful regulatory tasks. And so the suppression of these genes takes a variety of complex processes involving what are called Crab zinc finger proteins. Now, without diving into all of this in too much more detail, let me just bring you to our latest discovery, which again is a bit of serendipity where work we did coincided with work done by others. And we could see that the loss of function in one of these, or others have shown that a loss of function in one particular zinc finger protein, 808, causes something called pancreatic agenesis. And this is where the pancreas fills up, instead of with pancreatic beta cells, it fills up with liver cells, which of course are useless. And this causes profound neonatal diabetes. It's a very rare condition, but nonetheless, it tells us that this zinc finger protein is critical. And then we found that variations in the methylation rates of this finger protein and other control areas around it also have a similar effect. So that's really pointing the finger very closely at this. And what we think is that this is just going to be the very first example of probably many where methylation helps to control and regulate these jumping genes and make sure that they don't misbehave. And so if we can understand that, then I think we can really make a lot of progress in terms of ameliorating some of these very severe conditions that are developmental errors, but partly related by methylation.
Interesting. Have you guys published that yet?
Yes, that's out in a preprint version.
Okay, great. Just to let our listeners know. So I'm afraid we have to leave it here. I'm just going to make a short summary of this to make sure that I have it straight. So what you're saying, basically, is that the nutrition of the mother, and especially her B vitamin status around the time she conceives a baby, can affect the methylation processes during the very first few days of embryonic development, and that this actually has lifelong effects on the baby's future metabolism and health. And as nutritionists, we should be focused on the huge importance of optimizing nutrition for mothers to be.
Yep. Because we've found the effects on diabetes and obesity and cancers and so forth, we really think that this is going to be important. Now, just as an endpoint, you might ask the question, well, are we in a position already to make a recommendation for mothers to be?
That was my next question.
And now that's where we're really stuck because, and this is the disappointing bit about it, some of these things go in the wrong direction. So let's say we were to recommend giving much more folic acid, that would increase the methylation of a certain region, which would be beneficial in terms of health. But it might well have a reverse effect in terms of some other outcomes. So there's still a lot of research to do, but we will ultimately get to a position where this very basic discovery research is going to tell us important clues as to how we can optimize a mother's nutrition and in the pre-conceptional time so that we get a better, healthier baby for its whole life at the end of that process.
But just to simplify for now, what would you recommend for mothers to be? What is your recommendation as a nutritionist? I think you should close with that.
Yes, it's kind of apple pie, motherhood and apple pie that we always say, which is, a diverse diet, particularly, I think, around conception, that it's important to have a good intake of vegetables, especially green leafy vegetables, lots of folic acid. I mean, the folic acid story is a slightly different one, which we'll cover in a different podcast, that pre-conceptional folic acid is very important to reduce the risk of neural tube defects. But apart from that, so a healthy diet. But another critical thing is a healthy body weight. And again, we'll cover that in a different podcast, that the body weight of a mum, her body composition at the time of conception is really important.
Right. Thank you. Okay. So I think we'll leave it there. Thank you for being a guest on this podcast.
Thanks, Carla. That's perfect. Your summary was perfect. And I guess it remains for me to sign off in the usual way by encouraging you to share this episode if you've enjoyed it, and leave comments on LinkedIn or whichever platform you use to access it. And please do subscribe through the NNI website or the NNI 365 app to ensure you don't miss future offerings.
Thanks for listening to Milestones. Explore more expert insights on the Nestle Nutrition Institute website and follow us on our social channels. See you next time for another milestone in nutrition.